So today we are going to look at the detecting amino acids. So actually in the detection of amino acids, we need to know that generally in chemistry, in biochemistry, we have two tests. Two tests. A test generally in the biochemistry, we have what we call uh, qualitative tests. We have what we call qualitative tests. Qualitative tests, we have qualitative tests, and we have quantitative tests. What is qualitative tests? Qualitative test is just to detect the presence of a particular analyte. When we say analyte, it means what you want to detect. So a qualitative test is a test that you use in lab to check the presence or absence of a particular analyte. And then quantitative, like for example, let me just give you example of qualitative. In this group, we have Habiba Ibrahim Hassan. So assuming now one of classmates is now wanted to know whether the Farida Ibrahim attended these lectures or not. So to check whether Farida attended this lecture or not is what we call qualitative. So assuming now you are given a solution, you are given a solution. Like for example, generally in a hospital, you do this. If you have a patient, maybe you want to check whether the patient is having malaria or not. So to check the presence or absence of malaria in a particular patient is what we call qualitative test. But for the quantitative test is about quantity. You already know that, okay, that particular patient is having malaria, but you don't know to what extent. What is the parastemia level? What is the quantity of the parasite, maybe in 1 ml of the blood? So quantitative test is just to tell you how much of your analytes you have in a particular given sample. Assuming now, okay, now you want to check whether Farida attended this lecture. So you now check, yes, there is Farida. But you don't know at the same time how many of those Farida are from your class that attended this lecture. Then you now check how many how many Farida attended this lecture. So to know that, okay, the number of the Farida that attended this lecture is what you call quantitative. But to check whether the Farida attended or not is what you call qualitative. So in this lecture, we are starting with the detecting of amino acids. So how do we detect amino acids? So detecting amino acid is the detecting amino acid is what we call qualitative tests. So generally, we use ninhydrin. We use ninhydrin to detect the presence of an amino acid. So ninhydrin, and this is the structure of the ninhydrin. Ninhydrin is a bicyclic compound. It has two rings, and it is a classical reagent used for detecting amino acids. So we must use it in detecting amino acids. So as I said, ninhydrin is a classical reagent that is used for detecting an amino acid. And this reaction requires 2 to 5 minutes at 110 degrees centigrade and is sensitive at the nanomole level. So what does that mean? It means that when you want to actually test the presence of this amino acid, what you need to do, let's say this is your solution, or maybe this is uh, this is your test tube. And in this test tube, you have your solution. And in this solution, you want to check whether amino acid is there or not. So in this case, you need a nanomole. That is, you need a small quantity or small concentration, small amount of this menhydrin, and you add it inside this test tube. And when you do that, when you add first, you have your solution in test tube, and then you now measure nanomole of that menhydrin and you add it into your solution. And you now subject this to a temperature of 100 degrees centigrade. Maybe you have, let's say, you have your water bath. So you transfer this mixture from the test tube and you take it into the water bath that is actually boiling at a standard or at the temperature of 100 degrees centigrade. And you allow it to stand for 2 to 5 minutes. And then after you do that, then the next thing you use spectrophotometer. Spectrophotometer is an instrument that's used to detect the absorbance of a particular material. So now you have it here, and this is your amino acid, and this is your
Sorry,
anhydride.
sir. Sorry, sir. That photometer, sir. I don't get it.
Yes.
Sorry.
Okay, spectrophotometer is an instrument that is used to measure the absorbance of a particular analyte. Spectrophotometer is an instrument that is used to measure the absorbance of a particular spectrophotometer. Is an instrument that is used to measure the absorbance of a particular analyte. So in this case, our analyte is an amino
Thank you,
acid.
sir.
So as I said, after you now add a nanomole of your analyte, you add it into the test tube containing your solution, and then you now subject it to a temperature of 100 degrees centigrade, maybe in a water bath. Then after you do that, then the next thing you now allow it to stand for a temp or at a, to stand to 2 to 5 minutes. And then after you do that, then you now measure the absorbance of the product, the products, the product. You see here, how many anhydrin do you have? We have 2 anhydrin reagent. And you now react it with an amino acid. And at the end, you see this amino group. You see, this is the amino group. So this amino group linked these two anhydrin reagent to produce these complex color products. And the color of this product is purple. Then you now measure it as 570 nanometer. So you see at the end of this reaction, you have 3 products. Number 1, you have this. And then this is what, if you look at it from, if you have a basic knowledge of biochemistry, of organic chemistry, this is ethanol. So you have ethanol plus carbon dioxide plus this color complex. And then after you have this, then you now measure it at 570 nanometer. So the product from this pro is yellow and absorbed at 440 nanometer. So it means that if you now measure the absorbance at this as 570 nanometer and you are able to get the absorbance, that means that you have an amino acid. And if it did not absorb, and if it did not absorb at this, it means that the amino acid is not there. Why? Because if there is no amino acid, there will not be linkage between these two anhydrin reagents. So you see, it's the nitrogen from the amino acid that links the two anhydrin reagent that make it to absorb at this 570 nanometer, and the color is purple. So with this, ladies and gentlemen, you'll be able to understand that if there is no amino acid, there will not be formation of this product and there will not be absorbance at this. So that is the detection of amino acids